Yeah. Good afternoon, dear friends. So, in the morning, we have discussed some detail about the genetic code. That only we are going to discuss here in very detail. So, what is actually the genetic code? Genetic code is nothing but the sequence of nitrogenous bases present in the mRNA only. And these nitrogenous bases only determine what type of amino acid should be placed during the process of translation. It is giving a code. It is giving a message for a particular amino acid to be placed in a particular area during the process of translation. And always this code is a triplet code only. 3 continuous nitrogenous bases in the mRNA only decide what type of amino acid should be placed here, and it is what the code consists of 3 nitrogenous bases, and we call it as what the triplet codons. Already discussed some of the details about the genetic code in the last class. See, the process of translation required the transfer of genetic information from the polymer of nucleotide of RNA to the amino acid of protein. All the informations are directly coded from the DNA to the RNA. In RNA, all the information related to the protein is coded in the form of what the triplet codon only, and based on that only amino acids will be placed in a protein. It is found that change in the nucleotide sequence in the RNA change the amino acid sequence in the protein. If the sequence of the triplet codon or nitrogenous base is changed in the mRNA, based on that only protein will be synthesized. So naturally, amino acid sequence also will be changed. And the George Gamow, a physicist, argued that since there are only 4 bases, which are the 4 base pairs? Adenine, guanine, uracil, and adenine, guanine, uracil, and cytosine. See, only 4 nitrogenous base. If it is called for 20 amino acid, it is not possible to code 20 amino acid. If one nitrogenous base is coding for one amino acid, it is possible to code only for 4 amino acids because only 4 nitrogenous bases. If you combine 2 as what the genetic code and go for its combination, it is possible only to code 60 amino acids, 16 amino acids, 4 into 4, only 16 amino acids. But 20 amino acids are there. So he told that in order to code for the 20 amino acid, the code should made of 3 nucleotides. 3 nucleotides. 3 nucleotide means what are the possibility? How many this triplet codon can be prepared if we consider 3 nucleotide in a codon? So 4 into 4 into 4. That is 64 triplet codes are possible. And by using these 64 triplet codes only, these 20 amino acids are coded for the protein synthesis. If it is only 1, only 4 amino acids can be coded. If it is doublet, only 16 can be coded. But we have 20 amino acids in the cell cytoplasm, so required minimum 3 nucleotide as a code for a particular amino acid, and we call it as what the triplet codon only. And if codon is with the 3 nucleotide, it will generate 64 codon, 4 into 4 into 4. And he explained that codon is a triplet. It is not doublet or it is not single. It is triplet only. See, an Indian scientist, Hargobind Khorana, you may be heard about him. He got Nobel Prize and proved it by preparing an mRNA artificially in his laboratory. And later, that Marshall Nirenberg, a cell-free system for the protein synthesis, helped to find out the meaning of the triplet codon. He only found the meaning of all the triplet codon. And he prepared an enzyme, Severo occur. It's a polynucleotide polymerase only, and use this polymerase to prepare an artificial RNA with a different type of nucleotide, and he only produce what? An RNA outside and identify the meaning of all the triplet codons. Every triplet codon is having its own meaning. Meaning means what? What is it called for? Which amino acid? Right. That only he identified by preparing one RNA outside the laboratory by using what? This polymerase enzyme, that is Severo-Ochoa, this polymerase enzyme, and find out this what the meaning. And this is what the triplet codon, 64 triplet codons are there. And 3 letters coding for a triplet codon. And this is what the first position and second position, and it is what the third position, right? And here U, then U, then here U. This is the first one, U, and here U, right? Taken here. So 3-letter code only used for coding a particular amino acid. And this is what the triplet codon, 64 triplet codon, by considering 3 nitrogenous base as a single code. And you see here, all are triplet codes only. And here to code for the phenylalanine, we can find there are 2 type of this triplet codon, UUU for phenylalanine and UUC also for phenylalanine only. And see the leucine. Leucine also coded with the 2 UUA and UUG. These 2 triplet codons are for what? A single amino acid only. See the proline, there are 4 triplet codon here. And leucine, there are 1, 2, 3, 4, 5, 6 triplet codons are there for the leucine. And proline, 1, 2, 3, 4 triplet codons are there. And serine, 1, 2, 3, 4 triplet codons are there. That means one amino acid can be coded with many number of triplet codes, because amino acids are only 20, but triplet codons are 64 out of 64. I told you 3 are stop codon only. So only 61 meaningful triplet codon, and this 61 triplet codons are coding for only 20 amino acid. So naturally, one codon is for a particular amino acid, but this amino acid can be coded with many number of triplet codons. So this is what the triplet codon having 3 nitrogenous bases, that nearby 3 nitrogenous bases is the first position, second position, and third position. Then come to the salient features of genetic code, and this asked once in the board examination. Salient features of the genetic codes. As I told you, the codon is triplet, consists of 3 nitrogenous bases and 61 codes for amino acid. There are 61 triplet codon code for these 20 amino acids, and 3 codes, triplet codes, are not code for any amino acid, and hence they function as what? The stop codon only, or we call it as what? The terminating codons or nonsense codon. See, one codon code for only one amino acid. For example, AUG, it code only for one amino acid, and hence it is what? Unambiguous and very specific. Obviously, it is very clear and very specific. And here, UUU, it is only for phenylalanine, and CUU only for leucine, and ACU only for threonine, or CGU only for arginine, right? So this triplet codon is very specific, and it is very specific for a particular amino acids. Some amino acids are coded by more than one codon, triplet codon. Hence, the codon is what? Degenerate and typically abnormal. So only 20 amino acids, but there are 61 triplet codon. So naturally, one amino acid can be coded with many number of triplet codon. Here we can see the many number of triplet codon code for the proline, and threonine coded with this much triplet codon, whereas methionine only one triplet codon, that is always AUG. I told you in the morning, AUG is for the dual purpose. One is to initiate the process of translation, and another one is what code for methionine amino acids. Then codon is read in mRNA in a continuous fashion. The triplet codon, all the nitrogenous base coded in the mRNA very continuously. There is no punctuation, there is no full stop, there is no comma, there is no gap in between. And the code is nearly universal. Universal means the same code is there in every living organism, right from bacteria to this what the human being. And here, from bacteria to human being, this code is same. The triplet codon is same. For example, U. The triplet codon is code for phenylalanine in all the living organism, right from the bacteria to the human being. That is why I told you this code, triplet code, is what universal. Some exemptions are there, and that we found in the mitochondrial triplet codon and the triplet codon present in some of the protozoan. These are the exemptions. Otherwise, it is universal. Everywhere, the triplet codon is meant for a particular amino acid only. And another salient feature of the triplet codon genetic code is AUG, as I told you, having the dual function. What are the functions of the AUG? It is code for methionine, only one triplet codon for methionine, that is AUG only, and also act as what the start codon. It is for initiating the process of the translation, right? Then come to the mutation and genetic codes. Mutation means what? The changes happen in the genetic material of the triplet codon. So triplet codon only decide what type of amino acid will be placed in that particular place, particular area of the protein. If it is changed, even a point mutation means what? Mutation only a particular position, only in the specific area. Even that point mutation can also change the chemical nature of the protein produced. And sickle cell anemia is what a very good example for such a point mutation produce what a totally different proteins. And this is what a very healthy normal RBC. See the shape and its structure, and it is a normal RBC, red blood cells only, and it can flow through this blood vessel without any kinds of block, unrestricted blood flow only, provided these healthy normal red blood cells. And shape is like that only. But because of what the mutation, what happened, it produced what? An abnormal protein. And this abnormal protein production only lead to what? The sickle cell anemia. See the shape of what? This RBC. And whenever it flows through the blood vessel, it can settle somewhere and it can block the blood flow. And that only we call it as what? The sickle cell anemia disease. Sickle cell anemia, it is due to what? This mutation of the genetic code, only responsible for keeping these amino acid exactly in the place. And we will see that the differences. This is what the hemoglobin genome for producing the hemoglobin only. Hemoglobin A, it is what? The normal one. And there is a DNA sequence called as what? The gene to produce what? This hemoglobin protein. And this GAGCTZ, it is what? The two strands of DNA only, and that produce what? An mRNA. mRNA is what? GAG. So if it is GAG, that CTC only acting as a template, and this is what? The coding strand only. This one is coding strand. This is what? The template strand. And it is what? The 3' strand. And this one is what? The 5' strand. And it produces what? An mRNA. Just to copy the mRNA. So mRNA, this is template. So naturally, the mRNA consists of what? This triplet codon is exactly like coding strand only, GAG only. It is what? RNA, but it is what? The DNA only. And this mRNA, GAG, triplet codon, is code for the glutamic acid. Here you can see that what you can say, that sequence, that is GAG. See, GAG, it is code for here you can see GAG and G, then AG, CGAG, it is for the glutamine. So what the normal one. And suppose if it is defected because of what? The mutation. So what happened in the defected HBS? S means sickle cell only. And this gene is here. This is what the DNA. The correct sequence is GAG and CTZ. And because of the defect, what happened? The central one, that is what called as what? The point mutation. Instead of A, this will be T, and instead of T, this will be A only. And naturally, it produces what? mRNA through the process of transcription. Instead of GAG, it produces what? GUG only. But this GUG, see this GUG, and this is what? The G, then U, then GUG, and GUG, it is for valine only. GUG is for valine. So here you see in the sixth position of the amino acid in this protein, instead of what? The glutamine, this valine will be placed. What will happen if the sequence is changed? It will not able to perform its function well in a body of the individual. That is why such a defect comes in the hemoglobin, and it is not able to carry enough quantity of oxygen to the cells. And this problem only, we call it as what? The sickle cell anemia. Then come to another important topic, that is regulation of gene expression. See, every individual consists of many number of genes in their chromosomes. And most of the genes are not at all expressing. And some genes are expressed continuously in their body. And some genes are expressing only at the time of requirement. Some genes are continuously expressing, but whenever they are getting this material from outside, it stops expression. And some genes are silent, but whenever they want to produce that particular protein, that time only it expresses that particular protein. So such a type of regulation we can find in the expression of gene. And that only we call it as what? The gene expression. And gene expression can be regulated in different levels. These are the different levels of gene expression. Gene expression can be regulated in eukaryote in the transcriptional level. Transcriptional level, you know, formation of the primary transcript, that is what? The mRNA. So mRNA production can be stopped so that the production of what? This particular protein can be regulated. So in transcriptional level, it can be regulated. In processing level also, it can be regulated. That means go for transcription and produce produce what? The RNA, hnRNA. But this hnRNA never go for processing, so that it is not possible to go for synthesizing the protein. That capping, tailing, then splicing, all these things are required for make an hnRNA to convert it into mRNA. So in processing level, it can be regulated. Then even after processing, it can be regulated in the transportation of mRNA from the nucleus to the cytoplasm. To synthesize a protein, actually that processed mRNA should be taken to the cytoplasm, because machinery for synthesizing protein available only in the cell cytoplasm. So that can be stopped, so that naturally there will be no synthesis of protein. And even translation level also, it can be regulated. There transcription take place, then maturation take place, and this matured mRNA is taken to the cytoplasm. But in translation level, it can be regulated so that no production of that particular protein in the cell or in the individual. So genes in a cell are expressed to perform a particular functions. Gene present in the cell are expressed to perform a particular function. Example, enzyme called the beta-galactosidase enzyme, synthesized by the E. coli for catalyzing the hydrolysis of disaccharides called as what? Lactose into galactose and glucose. See, in case of E. coli, bacteria, gene is there to produce an enzyme called as what? The beta-galactosidase enzyme. And actually, this E. coli is synthesizing this beta-galactosidase enzyme by using this gene to digest what? Disaccharides. Two monomers are there in disaccharides called lactose. So, because of the digestion of this lactose by using the beta-galactosidase enzyme, this disaccharide lactose will be converted into the monosaccharide galactose and glucose. See, maybe heard about this lac-operon. Lac-operon is what? The regulation of the gene expression only. There is a gene in the body of the E. coli to produce what this enzyme called beta-galactosidase enzyme. But E. coli is not producing beta-galactosidase enzyme continuously in their body. See, whenever we are putting this E. coli into lactose medium, you know, its membrane is permeable, so naturally lactose will enter inside the body of this E. coli. So after taking this lactose in the body of E. coli, to digest this lactose into what the galactose and glucose, they should produce beta-galactosidase enzyme. So gene is there in their genome to produce the beta-galactosidase enzyme, and it produces it only after the entry of lactose in their body. Otherwise, no production of beta-galactosidase enzyme, and this gene is silent. If it is there in the lactose medium, if lactose is there inside the body of the E. coli, to digest the lactose, naturally it starts producing what? The beta-galactosidase enzyme, right? So come to the regulation of the genetic expression. As I told you, E. coli produce beta-galactosidase enzyme whenever it gets lactose from their living surrounding to digest it. If no lactose available in the living surrounding, E. coli never produce the beta-galactosidase enzyme. Even then it is having the gene inside, it never produce beta-galactosidase enzyme because it is not required, because it is only to digest the lactose. Always these enzymes are produced in the cell depend on its need or the demand of the enzyme by the cell. Whenever it required beta-galactosidase, that time only gene will express the character. Otherwise, gene will be very, very silent. And production of beta-galactosidase enzyme is under the control and regulation of a group of genes called as what? The operon genes. That concept only we are going to discuss here, that lac-operon concepts. So there is a gene in the genome of E. coli to secrete or to produce beta-galactosidase enzyme to produce what the to digest this lactose enzyme, lactose, but there is a group of genes present in the genome of E. coli to control or to regulate the expression of this gene, beta-galactosidase enzyme, and these genes are collectively called as what? The operon genes, right? And gene regulation in the individual mainly in two ways. One is a positive way of gene regulation, and another one is what? The negative way of gene regulation. What is the positive way of gene regulation? And this positive way of gene regulation is called as what? The induction. And negative way of gene regulation is called as what? The repression. So with the two examples, I can develop this negative and positive way of gene regulation. Just now I told you about this what? The E. coli. E. coli express the beta-galactosidase enzyme not continuously, only after the entry of the lactose inside the cell. And that is what the positive regulation only, or we call it as what the induction only. That means in induction, gene is not permitted to express continuously. It is not producing that enzyme continuously. Whenever it is required, that time only it go for production of the enzyme or this protein. And that is called as what the positive type of gene regulation or induction. And some cases, like what you can say, tryptophan production in the E. coli, it is continuous. There are some genes present in the E. coli continuously producing what the tryptophan. But whenever we are putting this E. coli into a tryptophan solution, because it is getting the tryptophan from outside, no need to prepare inside. So what happened? The gene stopped the production of the tryptophan inside. Continuously it is producing, but whenever it is getting from outside, it stops its production. That is what the negative way of gene regulation, and that only we call it as what the repression. Induction and repression, these are the two way of gene regulation. Induction is what the positive way of gene regulation, and repression is what the negative way of gene regulation. And two examples are also mentioned here. Lacoperon is an example for the positive type of regulation only. E. coli is not producing beta-galactosidase enzyme continuously in their body, but whenever it required, it start preparing. At what time it required this enzyme? Only after the entry of lactose inside. And such a regulation is called as what the positive type. And induction is called as what the induction. Whereas in tryptophan operon, is an example for the negative type of regulation only. And here, E. coli is continuously producing the tryptophan in their cell. But whenever we are putting the E. coli into the tryptophan medium, it is getting the tryptophan from outside continuously. So E. coli stop the production of the tryptophan inside. So this way of regulation of gene is called as what the negative type, and call it as what the repression only. And we have to study only that positive way of gene regulation. Regulation in the class 12 example is Lacoperon. So that only we are going to discuss here in detail, and it is very important. Last year also, I've seen one question from this operon, Lacoperon. So a very easy concept thing is that the diagram should be by hearted, so that very easy to explain it. So in Lacoperon, operon means what? A group of gene to regulate a particular gene to express a enzyme. So gene which is expressing an enzyme is called as what? The structural gene. Structural gene. Here we can find a group of genes in the Lacoperon system. I gene is there, promoter gene is there, operator gene is there, Z gene is there, Y gene is there, A gene is there. So there are many genes in this Lacoperon system to regulate the expression of what this enzyme called as what? The beta-galactosidase enzyme. Okay, and the gene which secrete that enzyme is called as what? The structural gene. And here there are three structural genes because it is polycistronic. What is polycistronic? This is what the what you can say genetic sequence which able to produce what many number of polypeptide chain. Here there are three type of polypeptide chain or protein produced by the structural gene. And it includes, there are three genes, Z gene, Y gene, and A gene. Z gene is responsible for the production of beta-galactosidase. Y gene is for producing the permease, and A gene is for producing what? The transacetylase enzyme. That beta-galactosidase, permease, and transacetylase enzyme. These three genes, these three enzymes or proteins are produced by the structural gene present in the Lacoperon. And this structural gene is operated by or regulated by another gene called as what? The operator gene. That only working here in the system as a switch. Whenever it is putting the switch on, operator put the switch on, naturally it make all the genes present in the structural gene, Z gene, Y gene, A gene, produce these enzymes together. And if operator put the switch off, what will happen? Z gene stop the production of beta galactosidase, Y gene stop the production of permease, and A gene stop the production of trans-acetylase enzyme, because all the three genes are operated by a single gene called as what? The operator gene only. And to control this operator gene, there are two more genes here. I gene is here and promoter gene is also there. And collectively, we call it as what? The log operon system. And whenever we are putting this E. coli into the lactose medium, so why this beta galactose permease and trans-acetylase produced by the structural gene? Because now the operator gene put the switch on. After putting the E. coli into the lactose medium, operator gene will be on. So naturally, Z gene, Y gene, and A gene express this character, express the proteins together. And this beta galactosidase enzyme produced by the Z gene present in the structural gene only convert the disaccharide lactose into the monosaccharide glucose and fructose. And this glucose and fructose actually used by this E. coli for energy purpose. They do what? Cellular respiration and liberate lot of ATP molecules. And this system only we call it as what? The lac operon. See here, you see the lac operon structural gene is regulated by a group of genes. Structural gene means what? The gene really express these proteins. Structural gene is a polycystonic, consists of three type of gene, Z, Y, and A. Z gene produce beta galactosidase enzyme, Y gene produce what? The permease enzyme, and gene A produce what? The trans-acetylase enzyme. Actually, permease enzyme only increasing the permeability of the membrane of E. coli so that lactose can easily enter inside. And these three genes are under the control of a single switch. Here, the operator gene only act as a switch. Whenever it put the switch on, all the three genes start expressing its what? The protein. And whenever it put the switch off, immediately all these three genes, Z, Y, and A genes, stop the production of the enzyme. And operator gene act as a switch in this lac operon to stop or to start the process of the system. Operator gene is under the control of inducer gene and promoter gene. That's what all the genes would work together to control or regulate the structural gene. And such type of group of gene help to control or regulate the gene expression is called as what? An operon system. It is in the case of what? Lactose digestion only. That is why we call it as what? The lac operon system. There is another system in the E. coli. We call it as what? The tryptophan operon system. It is, it is a negative way of regulation only. There is another system for operating that tryptophan production, that is called as what? The TRP, tryptophan operon system only. Then let us see how it's work. There are two things mentioned here. One is in the absence of what? Inducer, and another one is what? The presence of inducer. There are two diagrams. This is what the diagram given in your textbook. Here, which one is the inducer? Inducer is lactose only. In the absence of inducer means whenever we put this E. coli in a water, not in the lactose medium, lactose is absent, inducer is absent. So naturally, it never produces beta galactosidase enzyme. And this is what the operon system. Then I gene is producing a repressor mRNA through the process of transcription, because mRNA only helping for production of a protein. Remember the protein synthesis, transcription, translation. So this I gene produce what? A repressor mRNA. And by using this repressor mRNA, produce what? A protein called as what? The repressor protein, and it is very active. After its production, it will go and bind to the operator gene. Operator gene is what? The switch only, switch off this entire system. So because of this binding of the repressor protein on the operator gene, it put the switch off. So the entire system stopped its functioning. Now the switch is off due to the binding of repressor protein. It is very active one, bind to the operator. So naturally, all other genes present in the system, what you can say, stop its functioning. So no chance to produce any kinds of enzyme by Z, Y, or A gene. The structural gene is not at all expressing, and it is totally regulated, and it is stop its secretion. Now switch is off only. In the absence of inducer, what will happen if you put the E. coli in the lactose medium in the presence of inducer? Here, inducer is lactose only. So, as usual, I gene produce a repressor mRNA, and this repressor mRNA produce a repressor protein here. But now the E. coli is in the inducer medium or in the lactose medium only. So after the entry of lactose, it binds with the repressor protein here and inactivate the repressor protein. That inducer is what? The lactose only, that bind to the repressor protein and inactivate the repressor, so it never go and bind to the operator. It never go and bind to the operator because it get inactivated due to the binding of the inducer lactose in the repressor protein. You see the diagram here. Now operator is not off, it is on only. So what will happen? It produces mRNA and protein, and it is stimulating that P gene, promoter gene. Promoter gene produce what? The mRNA and the protein that will stimulate the structural gene Z, Y, and A. So Z, Y, A structural gene produce what? The lac mRNA, and by using this lac mRNA, it go for translation and produce three type of enzyme: beta-galactosidase enzyme, permease enzyme, and transacylase enzyme. And by using this beta-galactosidase enzyme, this inducer enter in the body, go for what you can say hydrolysis or go for digestion. The disaccharide will be converted into monosaccharides. So this is what a regulating system present in the body of E. coli to control or to regulate the expression of the gene to produce these three enzymes: beta-galactosidase, permease, and transacylase enzyme. It is what? A positive way of regulation only, because E. coli produce and express this beta-galactosidase enzyme only at the time of need. Whenever lactose enter in the body of the E. coli, that will bind to the repressor and make it inactive, so that it never bind to the operator, so that this operator put the switch on, so entire structure start functioning. And finally, what happened? It induced all the genes of the structural gene, and ZYA genes start expressing what? Their respective mRNA, and through the process of translation, this mRNA will produce what? Three type of protein: beta-galactosidase enzyme, permease, and transacetylase. These are all the enzymes, basically protein only, and digest what? These lactose into its monosaccharides. Right? So this diagram you should be by-hearted, so that it is very easy to explain the concepts. The role of what the permease is to increase the permeability of the membrane of this E. coli, so that outside, from the outside, because of the difference in their concentration, lactose can easily enter inside the cytoplasm of E. coli. So this is what in the presence of inducer. See how this operant system regulate the expression. If it is there, if inducer is there, this operator system, I mean this system is very active, and operant system is very active and start functioning. If inducer is not there, because of binding of the repressor on this operator gene, it put the switch off and everything is silent. Right, that is what the regulation, positive type of regulation. And that's all about what the regulation of gene expression. And the next topic is what the Human Genome Project solely. I think it is an easy topic. You can read it. Actually, this project is to identify the sequence of nitrogenous base present in the human being in all the chromosomes, 23 pairs of chromosomes. It's very costly, required a lot of time and support from many countries to do it. And just to go through what the goal of the Human Genome Project, and here there are two methods to do the Human Genome Project. One is what the expressed sequence tag, expressed sequence tag. And here we identify the sequence, nitrogenous base sequence of the gene that express as RNA to synthesize a protein. As I told you, many genes are very silent in our body. Many genes are very silent, never it produce RNA, never it produce what the protein. And here we are trying to identify the sequence of the gene which is expressing a particular protein by producing an RNA. That is what the expressed sequence tag. Then another one is what the sequence annotation. In sequence annotation, identify all the sequence of the DNA, irrespective of their function. Some are functional and some are non-functional. Some are producing protein, some are not at all producing protein. We are sequence all the nitrogenous base sequence from one end of the DNA to another end. That is what the sequence annotation, irrespective of its function. It may be functional or it may not be functional. Of course, it is having lot of application, methodology, simple one only. Then salient features of Human Genome Project is given many numbers, right? And salient features of Human Genome Project, go through it. Chances are there to ask many questions from this area. Then another important topic. Why I skip all these things? Because time limitation is there. But it is important at the same time, very easy topically. By reading, you can understand. So concept of Human Genome Project is to sequence all the base, I mean nitrogenous base sequence present in our DNA from one end to another end. Then come to DNA fingerprinting. It is very important topic. DNA fingerprinting. So it is not like what you can say thumbprint. No, it is very unique. These people, those who are investigating the crime, used to take the fingerprint from the scene of crime. That is to identify the person involved in this crime because it is very unique. Similarly, our DNA is also having its own, what you can say, identity. And that only we call it as what the DNA fingerprinting. It is not the fingerprint, but it is what the sequence of the DNA. It is just like what our thumbprint, it is very, very specific. And 99.9% of the base sequence in the human being is same. That is why we all look alike. 99% of sequence of the human being is same and it is helping to perform all the basic functions in the body. Of course, we can find a lot of differences in our body externally in the form of color or shape or height or something. That is what you can say only because of what the 0.1% of genetic difference. And this 0.1 genetic difference make every individual unique in their phenotypic appearance. But internal, what you can say, functioning and all other things are same. That only because of what 99.9% of the genetic similarity. So it is not easy to find out a total sequence of the individual to know the genetic difference. As I told you, while discussing about the Human Genome Project, it's very costly, required a lot of time. It is very difficult to sequence an entire length of the DNA from one end to another and analyze, compare these two persons to identify it. It is not possible. And it is very expensive only to trace 3 into 10 raised to 6 base sequences of two individuals to compare. So DNA fingerprinting is very quick way to compare the DNA sequence of any two individuals. So in case of dispute, we can identify the real biological father through the DNA fingerprinting. Actually, this DNA fingerprinting involves identifying the differences in some specific region in the DNA, not the entire length of DNA. We are taking only some specific region where the same sequence repeat for many times. And we call it as what? The repetitive sequence of DNA. G is repeating for 100,000 times. Or what you can say, particular sequence, A, C, Z, or like that. This sequence is repeated for many times, but not having any function in the body. They are not coding for any protein, but still it is there. It occupies what most of the areas of our chromosome, and it is very much necessary for determining the structure, but it is not at all coding for any kinds of enzyme. The repetitive sequence of the DNA, that only we used for analyzing the genetic difference between or similarity between these two individuals. Right? And here, repetitive DNA is the sequence of DNA repeated many times. 100 times or 1,000 times. This repeated sequence of DNA strand can be separated from the bulk of genome DNA by using density gradient centrifugation, and we call it as what? The satellite DNA only. In a DNA sequence, we can find in many places, in all the chromosome, all the 23 chromosome, we can find such type of what? The repeated sequence of DNA not having any kinds of what you can say functions to produce a particular protein. That only we call it as what? The satellite DNA, and this can be categorized according to what? The nature. Some repeated sequences are rich in AT, adenine thymine rich area, and some repeated sequences are rich in GC, guanine, and cytosine. And length of the segment, number of repetitive units, then the satellite DNA can be classified in many ways into microsatellite, mini satellites, etc. And this repeated sequence never code for any type of protein, never go for transcription, never go for translation, but they form a large portion of the human gene. In every chromosome, we can find such type of repeated sequences, and it's not having any function, not at all going for transcription or translation. And this repeated sequence show high degree of polymorphism. What is polymorphism? Polymorphism means what? The differences. That only we are using here to identify a person. If it is same in all individual, it is not possible. And it is showing what? The high degree of polymorphism. Repeated sequence of all the chromosomes are not same in every individual. And this difference never make any kinds of differences in the phenotype of the individual. Why? Because it is not at all coding for any kinds of protein. It is what? Actually the functionless, only the repeated sequences of the DNA. It is there. Bulk of such repeated sequences are there in every chromosome, and it is not at all creating any kinds of physical variation, but chemically this is different from one individual to individual. At the same time, it can be inherited also from one generation to other generation. So that is why we are able to find out the real biological father through this, what you can say, DNA fingerprinting. It can be inherited. So in the last examination, one question asked here, they are showing the bands of what you can say fingerprinting taken by from a father, a biological father, and a child. They are giving what the 3-4 options of father, but that 50% of the band of the child you have to see, and you have to identify in which suspected person this band is there. That 50% band exactly present in the child, because 50% of the genetic contribution from the paternal side only. So we can easily identify the real biological father through this DNA fingerprinting techniques, right? Then polymorphism of repeated sequence. Polymorphism means what? The differences in the chemical nature of the repeated sequence are inheritable from parents to children. So DNA fingerprinting can be used to identify the paternity in case of disputes. Polymorphism is a genetic variation in DNA sequence only and arise due to what the mutation only. And these repeated sequence are not affecting what the characteristic features of the individual, even then it go for mutation. Why? Because the repeated sequences are not at all coding for any kinds of protein. So if anything happened in the chemical nature, it will not affect the function of the individual. If an inheritable mutation is observed in a population, high frequency referred as the DNA polymorphism, because the DNA polymorphism is what the chemical changes in the sequence of what this nitrogenous base in this repeated sequence, that will be very high if it undergo the process of mutation. And polymorphism is greater in non-coding sequence of DNA. Non-coding sequence, it is not coding for any kinds of protein, which can be used as a tool for DNA fingerprinting. This will not affect the body or reproductive ability of the individual, because these are all non-coding sequence only. And this technique, DNA fingerprinting, actually was developed by the Alec Jeffrey only, not for identifying the criminal, but to understand the genetic defect in the early stage for very effective treatment. But now this is used mainly for identifying such type of criminals only. And he used the satellite DNA, showed a high degree of polymorphism. This, what you can say, the scientist Alexander Jeffrey, he used the satellite DNA only because this satellite DNA shows what the high degree of chemical changes or the polymorphism, and he called this, what you can say, polymorphism or the chemical change in the satellite DNA as what? Variable number of random repeat, because these are all repeated sequence of what particular nitrogenous base phase only for 100 times or 1,000 times. And he called it as what? VNDRS. Once it's asked in the board examination, expand VNDRS, that is variable number of random repeat. Nothing but the repeated sequence of nitrogenous base present in the DNA of a chromosome, and not coding for any kinds of enzyme. It is not coding for any kinds of protein, but it is there, and it is showing high degree of polymorphism or what you can say, chemical variations. That area only commonly used for this. Then come to the steps involved in the DNA fingerprinting. Now we are discussing about the various steps in the process of what the DNA fingerprinting. Isolation of DNA. See that from the scene of crime, the investigators used to collect these biological materials, maybe in the form of what? The blood sample, or maybe in the form of that semen, or it may be in the form of the hair follicles or what? The epithelial cells, something they left in the scene of crime. They collect and isolate the DNA from such a cell. They get only very limited number of cells from the scene of crime, but they can multiply the genetic content DNA without changing its chemical nature. Maybe heard about the techniques outside the cell, the genetic content can be multiplied. Right? Then digestion of the DNA by using restriction endonuclease enzyme. Here digestion means what? Converting the lengthy DNA into small fragments. That is what the digestion, fragmentation. How to convert this very lengthy DNA into number of fragments? By using endonuclease enzyme. Biological scissors, maybe heard about it. Biological glue, as we discussed, ligase enzyme only. This is what the biological scissors to cut the DNA molecule. And it is what the restriction endonuclease only. There are two types of restriction enzyme. One is endonuclease and another one is exonuclease. You know the differences. Exonuclease cut the DNA only at the end by adding methyl group. Whereas if you want to cut the DNA, according to our desire in between, it is possible only through the endonuclease. That is why we are using only the endonuclease for the DNA fingerprinting techniques. And after cutting this very lengthy DNA into small fragments by using endonuclease enzyme, all the fragments should be separated. How to separate the DNA fragment? By using the electrophoresis. We will see in detail about all these steps one by one. Then, that electrophoresis machine is able to separate the DNA fragment according to the length of the DNA. And after separation, this DNA, we have to convert the double-stranded DNA into single-stranded DNA in the electrophoresis machine itself. In the gel itself, we have to treat it with alkaline material and convert the double-stranded DNA into single-stranded after separating the fragment. Then we have to press a nylon sheet on the gel, or we can use the nitrocellulose sheet on the gel and just blot it. And that only we call it as the blotting process. Just to press it gently. So from the gel, this separated the single-stranded DNA stick to the sheet, nitrocellulose sheet or this nylon sheet. Then go for hybridization. Now it is single-stranded DNA only. At this single-stranded DNA, go for hybridization. Make it what you can say that complementary new strand. And hybridite is why it's called as hybrid, because one strand is non-radioactive and newly constructed one is the radioactive. So hybridize this VNDR probe. Hybrid it is with the VNDR probe and put it into the autoradiographic machine and detect this hybrid DNA, and it show a lot of band because it emit light that will appear in the X-ray sheet in the form of band. So by seeing this band, we will able to identify or able to compare this VNDR or this VNDR of two different persons, right? So this is what the different steps involved in the process of DNA fingerprinting. Then first one is the isolation of DNA. See, after getting a cell from the scene of crime, we have to make the DNA fingerprint with that. We have to isolate the DNA present in the nucleus of that particular cell. How to isolate it? How to isolate it? We need only DNA, not RNA, protein, carbohydrates, or lipids. So we have to treat the cell with all type of digestive enzyme, except DNases. If you treat it with the DNase, naturally DNA also will digest. We need only DNA. So treat it with the pectinase, so pectin will be digested. You treat it with carbohydrates, carbohydrate will be digested, and treat it with the protein, protein will be digested. Treat it with the lipids, lipid will be digested, and treat it with RNase, RNA will be digested, and treat it with cellulase, cellulose also will be digested. So pectin, cellulose, then RNA, lipids, carbohydrate, protein, then everything can be digested and can be removed. And after treating the cell with all this digestive enzyme, last chromosome will, DNA will be there in the solution. That can be precipitated by using what? A chilled purified alcohol. That DNA only left after treating the cell with what? All kinds of digestive enzyme, and this purified DNA can be isolated from the cell by using what? This chilled ethyl alcohol, and this can be collected in the form of what? Very fine threads. You may be conducted the same experiment in your school, that extraction of DNA from the peel of banana, or extraction of DNA from apple, or extraction of DNA from what? The vegetable peel like a tomato, right? It is very possible we are taking what? The content cell from this plant part, grind it with what you can say all kinds of digestive enzyme except DNA. Normally, we are using what? The cleaning solution only. In school, it is not possible to get everything. So instead, we use what you can say the dishwash. Dishwash consists of what? Mostly proteases, carbohydrate, lipases, cellulase, and pectinase, etc. DNA is not there. So we can digest what? All the unwanted thing present in the cell, and later it can be treated. We can pour the chilled alcohol, so we will get what? The DNA in the form of small fine threads that can be spun it out. This is what the isolation of DNA. And after isolating this DNA, right, we can cut the DNA into small pieces by using restriction endonuclease enzyme. I told you restriction enzyme only to cut the DNA. It is called as what? The biological scissors. There are two types of restriction enzyme. One is exonuclease and another one is endonuclease. But exonuclease cannot be utilized here for DNA fingerprinting because it cut the DNA only at the end. But we have to make the DNA into many fragments. So here we have to use endonuclease enzyme only. Maybe heard about the palindronic sequence and other terms. Every endonuclease enzyme, there is a 6 sequence. We call it as what? The palindronic sequence, because we can read in both direction in the same way. Right. That is what the palindronic sequence. Then the palindronic sequence of one endonuclease is different from another one. A specific palindronic sequence is there for a particular endonuclease enzyme. So by using that restriction endonuclease enzyme, we can cut the DNA in a particular sequence. As I told you, exonuclease is there, endonuclease is there, but exonuclease we are not using. Only endonuclease enzymes to cut the DNA between the DNA strand and make different fragments of the DNA and of different sequence. These restriction endonuclease are commonly used in the DNA fingerprinting technology. This is what the restriction. This is a long chain of what? Chain of nucleotide only, this DNA here. And if you want to cut the DNA in between, we have to use only what? The restriction endonuclease enzyme. So it is having its own, what you can say, palindronic sequence. It identifies the palindronic sequence and it cut it from both sides, little away from the center, you may be heard about, and it is producing what? Such a type of sticky ends. Whenever we pour this enzyme in the bulk of DNA, wherever they found their sequence, palindronic sequence, they will cut and make into number of small pieces of DNA. That is the second step, fragmentation of the DNA. Then next one is what? The separation of DNA fragment by using electrophoresis. After cutting this DNA into many fragments, we have to separate these DNA fragments. How we will separate it? By using electrophoresis only. So this is what the electrophoresis machine. And here we are using what? A gel for the movement. That is called as what? The agarose gel only. This is what? The agarose gel. And in negative electrode side, we can find so many sample wells, right, where we are keeping all what the DNA fragments. Some are very lengthy, some are short, because randomly only we cut the DNA by using the restriction endonuclease enzyme, and we place all the samples of the DNA in the wells present in the electrodes, negative electrodes. Then this is what the positive electrodes in the another end, and we are keeping it in the negative electrodes only, all these fragments of DNA. And DNA is basically negative in charge, so it is having a tendency to move towards what the positive electrodes. And the movement of this DNA fragment from the negative to the positive depends on what the size of the fragments only. So it start moving through the agarose gel only. It is moving from negative side to the positive side, and whenever the power is turned on and DNA fragment migrate through the gel, this is what the agarose gel, it is providing a medium for the movement from negative to positive. It start moving from the well. These are the wells in the negative side, and from the well it start moving. But these the small fragments of DNA can move away, whereas the largest one move, but very, very slowly only. So we are separating the DNA fragment based on the length. The small one move so quickly through the medium, and a large one move very slowly by taking time. So after what you can say, running this electrophoresis for a period of time, we can find the separation of the DNA fragment according to the length. Smallest one will be near to the positive charge, and the largest one will be there near to the pond or near to the negative electrodes. This is what the way through which we are separating the DNA fragment according to the size. The DNA fragments are separated by the size only. And it is called as what? The electrophoresis, right? So this is what the separation of DNA fragment by using electrophoresis. Fragments of DNA can be separated by using gel electrophoresis only, and gel only used here. What type of gel is used in the electrophoresis here? Agarose gel, it is what? A natural polymer extracted from the seaweed. From the seaweed only we extract the agarose gel, and through this agarose gel only this DNA is moving. And this provides the medium for the movement of the DNA in the electrophoresis. And DNA molecules are, negatively in charge, and which will be placed in the well located in the negative charge called the cathode end only. And this negatively charged DNA move towards the anode, having the positive charge under an electric field through the gel called as what? The agarose gel. And the DNA fragments can be separated according to their size. Smaller fragments move away quickly to the positive charge, whereas the large one will be near to the well in the negative side only. And this is what the way through which we separate this DNA according to the length. Now, DNA is separated. You can see the separated DNA here. Separated DNA is here. Largest one will be here, and the smallest one will be here near to the positive charge. And this DNA is double-stranded only. Then next aim is to make it what you can say single-stranded by denaturation. This is what double strand, it is what the real nature that we have to denature by breaking the hydrogen bond. We will apply some alkaline medium. In this alkaline medium, double-stranded DNA will be converted into single-stranded DNA. After the application of this alkaline, in this agarose gel, this only the single-stranded DNA appears in the gel, and this we have to blot it. We have to blot it, and it is only, we call it as what? The southern blotting. And southern blotting is like this only. These are the separated DNA fragments, and it is single-stranded only. And we will take the nitrocellulose sheet, or what you can say, this nylon sheet, nitrocellulose sheet, we will take here, and just we will place it on the gel, and gently we will press it. Gently. We press it on the surface of the gel. So the separated single-stranded DNA present or scattered on the gel, what you can say, attached to the gel like this. And this process only we call it as for the Southern blotting. We just blot the separated DNA on what you can say the nitrocellulose sheet. So these are all single-stranded DNA, and it is just taken from what you can say taken from the agarose gel through a simple gentle pressing only. And this process only we call it as what the Southern blotting. So this is the process of transfer of separated DNA fragments to the synthetic membrane, such as what the nylon sheet or the nitrocellulose sheet. And this is done by the gentle pressing of nitrocellulose or nylon sheet on the agarose gel present in the electrophoresis. Gently we have to press. The complete DNA fragments after separation in the electrophoresis can be shifted as it is to the sheet, as it is, without changing the location after pressing. This will be stick on the nylon sheet or the nitrocellulose sheet. But this is done only after making double-stranded DNA present on the agarose gel into single-stranded by treating it with what the alkaline solution only. Now we can see on the sheet, nitrocellulose sheet or the nylon sheet, the single-stranded separated DNA, and alkaline solution only we are using for converting the double-stranded DNA into single-stranded DNA, and finally single-stranded DNA fragments attached to the nitrocellulose or this nylon sheet through the process of what the Southern blotting. Then go for hybridization. Now DNA is single-stranded. It is attached to the nitrocellulose sheet or the nylon sheet. It's separated in different places. It is attached. Then we have to hybridize it. Hybridization means what? The conversion of the single-stranded DNA into double-stranded DNA. But in hybrid, there are two strands. One strand is what the old strand that we just blotted out from what you can say that agarose gel only. Then another strand we have to construct artificially by using the radioactive probe. And one strand is what made with the radioactive probe. It is radioisotope of this only. And another one is what the normal one. That is why we call it as what hybrid. One is ordinary strand and another one is what the light emitting strand only. Artificially synthesized. See, make this single-stranded DNA present in the nitrocellulose sheet into the double-stranded. It can be done by hybridizing the single-stranded DNA with a radioactive probe of nucleotide. It is making what? It's on complementary strand. That old one is just like what is a master strand only, and it is making a complementary strand, but by using what? The nucleotide, radioactive probe of nucleotide only used here for the hybridization. And after hybridization, the newly constructed DNA strand, double-stranded only, getting the radioactivity. The process of making single-stranded DNA into double-stranded DNA is called as what? The hybridization process. Here, no strand is radioactive because it is made with the what you can say radioactive probe of nucleotide, and old one is what? The non-radioactive. And this only hybridization. This is what the old one, and this is what the newly constructed DNA, and it is made with the radioactive probe only. All the nucleotides are radioactive probe only, and this is called as what? The hybrid. This is what the double-stranded DNA converted into single-stranded, and this single-stranded DNA go for hybridization, making a double strand with the help of the radioactive nucleotide and produce what? A hybrid. Why it is called a hybrid? One is with the non-radioactive nucleotide and another one with the radioactive nucleotides only. And this is called the hybridization technique. Then after hybridization, now it is there in the nitrocellulose sheet only. And next, we have to go for the autoradiography. Autoradiography means what? Expose this DNA to the X-ray sheet. An X-ray sheet is very sensitive to the light. Just to keep the X-ray sheet on its surface, because radioactive, this probe is emitting light all the time, so it can create lot of markings on the X-ray sheet. The radiation emitted by the DNA created marking on the X-ray sheet. This marking varies according to the length of the DNA, or as well as what the sequence of nitrogenous base present in the DNA. Marking will be different, and this process is called as what the autoradiography, and this will produce different type of banding pattern in X-ray sheets. Whenever we expose the hybrid on an X-ray sheet, because of what the emitting of light continuously from it, it create what lot of banding pattern, and this marking or banding depend on the length of the DNA strand as well as what the sequence of nitrogenous present in the DNA strand only. So like we will get what many bands on the X-ray sheets. Many bands we will get. It is because of what the light ray emitted by the radioactive what you can say probes present in the one DNA strand only. These are all what the markings appearing on the X-ray sheet is called as what the autoradiography, right? Then just to see the compare, this is what the fingerprint taken from fingerprint of Hazel taken from the scene of crime. You can see the banding pattern here, banding pattern. After investigation, normally they suspect many people. This is what the suspected person number 1, suspected person number 2, and suspected person number 3. Then they will take the fingerprinting of all the suspected person and compare the fingerprinting of all the suspected person with what the fingerprint prepared from the cell collector from the scene of crime. See, you can find, after comparison, you can easily identify which suspected person is really involved in the crime. See, compare it. Is it suspected person, number 1, or it is number 2, or it is number 3? So, see the banding. This band you can find only in the suspected person number 2. And this band is there in number 1, number 2. And it is there in only number 2 and number 3. And this band present only in number 2. And this band is there in number 1 and number 2. And this band is there in number 1 and number 2. And here this band is there in suspected person number 1 and number 2. That means all the bands appears in the fingerprint of the cell collected from the scene of crime can be seen in the suspected person. So the suspected person is there. Suspected person number 2 is there in the scene of crime or at the time of crime. This is what the scientific way of investigation. And here, instead of, in case of dispute, we can identify the real biological father. Sometimes confusions will be there or conflicts will be there. So that can be proved by what you can say scientifically by taking the fingerprinting. We have to take the fingerprint of the child, and we have to take the fingerprint of what the persons. Then what you can say, then compare it. Compare the fingerprint of this child and fingerprint of this person to find out whether that person is really the biological father of the child. And the child is having what number of bands. It will not appear 100% in this individual if he is a real biological father, but 50% of the band present in the child will appear in the, what you can say, fingerprinting of this person if he is what the real biological father. And 50%, that mother band totally. So here we can see another one, another comparison here. This diagram only given in your textbook. This is what the DNA from the crime scene. See, DNA from crime scene. There are 3 samples have collected for the DNA fingerprinting, and this is what the crime, I mean DNA from the crime scene. It's not only one. He has taken what 3 chromosomes. Chromosome number 11, chromosome number 7, then chromosome number 2, and chromosome number 16. See, every chromosome consists of such type of repeated sequence of what DNA. We call it as what variable number of tandem repeats. See, whenever we compare this variable number of tandem repeats, we have to take the variable number of tandem repeats only from the same chromosome. Here, from this, what you can say, the crime scene, taken the SL and consider 3 chromosomes, 7, 2, and 16. Same chromosome only taken here also. This suspected person, individual A, another suspected person, individual B. Same chromosome, 7 taken and 2 taken, and repeated VNDRS of 16 also taken for comparison. So it will be more valid. Then prepare the this fingerprinting of what the cell collected from the scene of crime. See, this is what the sequence. Then this is what the what you can say fingerprint of the suspected person, individual A, and this is what the second suspected person, individual B, fingerprinting of these 2 persons. And from 3 chromosomes, chromosome 7, chromosome 2, and chromosome 16. It is given in 3 different colors, red, yellow, and blue. And compare it with what you can say, the fingerprint of the cell collected from the scene of crime. Just to compare it, you just compare it with what you can say, these 2 suspected individual A and B, and this is C is what the DNA taken from the scene of crime, and compare it. And these red patches, you know, it is from the chromosome 7 here, chromosome 7, and blue one from the chromosome 16, and yellow one is from the chromosome 7. And just to compare it with this, here we can see the same patches, whereas in this person, individual A, and its band is here only. And second red band, you can find it here. And yellow band exactly here. Of course, this person is also having the yellow band here, but you just to see all the bands and compare. And this one exactly here, and blue band here, and both are having, but it is what you can say, more here in the B individual, and here this is what another blue band, and here is another one. That means this suspected individual B show 100% banding similarity with what the fingerprinting of what the cell collected from the scene of crime. Means what? That person is there at the time of crime, and if this person is also there, he also involved in the crime. So this is what the way through which we investigate the crime scientifically by using what the fingerprinting. So this fingerprinting is also what very important topic only. So all the process of this fingerprinting, you have to go through it and by heart the process of fingerprinting. So this is what the last topic. 1 minute. Sir. Sir, how much time, sir? How much time? 5 more minutes, sir. Can stop, huh? Okay, this is what the end of this chapter, last topic, and this topic is very important, right? And 5 more minutes. So if you want, we will go for discussing this topic that you omitted while coming. And this is what the Human Genome Project only. If you want to discuss, we can have it. Otherwise, there is simple topic only. And just to go through the goals of the Human Genome Project, methodology, and its importance. Okay, and we will see another topic in the evening. So taken from genetic engineering only. So we will see the topic after your break, and it start at 6:30 to 8 o'clock. So we will see some important area of genetic engineering. And first we will see the application of genetic engineering, because that principle and process, take a lot of times to complete. And we will see that uses of genetic application of genetic engineering. So all of you go for your tea and come back at 6:30 and join with me for the next section. Okay, thank you. Thank you so much.